check, 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 check. Check, 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 check. Mic check, 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 check. Check, check, check. Check, check, check. Check. Check, 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 check. Check, check, check. Check, check, check. Check, 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 check. check, 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 mic check, mic check, check, mic check, 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 check. Today's topic is genetic engineering. Check. தூரத்தில் கேளு நான் பேசுறது கேட்குதாரு. Very good evening, children. Today's topic we are going to talk about genetic engineering. Today's topic is genetic engineering. We'll start the class in another 10 minutes time. So children, be ready. Today's topic is genetic engineering. In this topic, we're going to talk about what are the various steps that are involved in genetic engineering process, methods of gene transfer, and then process of recombinant DNA technology, how the process is done, and then finally we're going to talk about the downstream processing. This is what today's class, and then we are going to talk about the applications of biotechnology. Okay children, we'll start in another 10 minutes time. Very good evening, children. We'll start our today's class. Today's class, the topic is genetic engineering. We're going to talk about genetic engineering and how actually the process is done, the various steps involved in that. All those things we are going to learn today. So let us start the class. Today's topic is about genetic engineering. Okay. So first let me define what is genetic engineering. Let us see what is genetic engineering. Genetic engineering, it is a process. It's all about modification of genes from an organism. How genes can be modified? Genes can be modified by altering the DNA. So here the genetic engineering means modification and transfer of DNA from one organism to another. That is called as genetic engineering. Genetic engineering is also known as recombinant DNA technology or rDNA technology. Since we are altering the genes, it is also known as gene cloning. It's also known as gene cloning process. It is actually a gene manipulation technique. This rDNA technology or genetic engineering involves the transfer of DNA, coding for a specific gene from one organism into another organism using a suitable vector. If suppose in a particular organism the gene is defective, those defective gene can be taken out and normal gene can be inserted. The insertion of normal gene, it is done through vectors. So this is known as recombinant DNA technology. And there are various methods are involved in transfer of the genes. Some of the methods are direct methods, some of them are indirect methods. The indirect methods like electroporation method, gene gun method, liposome mediated method, chemical mediated method, and micro injections. Whereas the direct method, it includes the TI plasmid, tumor inducing plasmids from Agrobacterium tumefaciens. Next we are going to talk about what are the steps involved in recombinant DNA technology. The first step that is involved in recombinant DNA technology, it is isolation of the DNA fragment. Here we have to isolate DNA from two things. One is the gene of interest, the gene that has to be transferred, that is called gene of interest. Similarly, the next thing is the plasmid. What is plasmid? Plasmid is a vector through which the genes will be transferred from one organism to another organisms. So here you can see that the gene of interest from a particular organism, it will be isolated and that it will be inserted into the plasmid. Here we have to cut the gene of interest from one organism. Similarly, we have to cut the fragment of plasmid, so that this gene of interest, it will be inserted into the plasmid. After the insertion of gene of interest into the plasmid, this plasmid, it is known as recombinant plasmid. Recombinant plasmid. Usually for plasmid, we will take the bacteria, E. coli bacterium. In the E. coli bacterium, it is a prokaryotic organism. This eukaryotic bacterium, it has two fragments. It has two nucleic materials. One is the nucleoid. This is the actual nuclei for bacteria. And apart from that, in the cytoplasm, there will be an extra chromosomal material. That is a circular extra chromosomal material. It will be present in the plasmid. It will present the bacteria. That is called as plasmid. So this plasmid, it is used as a transferring agent. So this plasmid, it is an extra chromosomal material, a circular extra chromosomal material, a double-stranded extra chromosomal material that is present in the cytoplasm of E. coli bacterium. So that is the most suitable vector for transferring genes. And now also we have one more vector, that is called the TA plasmids, which are widely used to transfer genes into the plants. Here, then, this is the process. This is the first step: isolation and generation of recombinant DNA. This is called recombinant DNA. Next is the DNA of the vector. It will be cut into fragment using a particular enzyme called restriction endonuclease. So this restriction endonuclease, it is an enzyme that is used to cut the DNA at a specific site. Restriction endonuclease. There are two types of enzymes are there: restriction endonuclease, restriction exonuclease. These are the one which is used to cut the DNA at a specific fragment, at a specific site. So with the help of that restriction endonuclease enzyme, first we have to cut the gene of interest. Similarly, according to that, we have to cut the DNA, or the plasmid from the E. coli bacterium. With the help of restriction endonuclease enzyme, we have to cut the fragment of both the gene of interest as well as the plasmid. And finally, the plasmid and the gene of interest, it will be joined by using an enzyme called DNA ligase enzyme, because that is the enzyme which links the DNA ligase enzyme. It's called paste enzyme. So this process of joining the gene of interest and the plasmid, it is called as splicing. So after splicing, what will happen? A recombinant DNA is obtained. Recombinant DNA is a combination of gene of interest and the plasmid. Next is once the recombinant DNA is obtained, it will be inserted into the host cell for further manipulation or further multiplications. The rDNA, recombinant DNA, it is introduced into the host cells. So there are basically three host cells we are using. One is the E. coli bacterium itself, the other one is Bacillus subtilis and Streptomyces species. They are using it as the host cells. Now what will happen? The recombinant DNA, it will be inserted into the host cells. So within the host cell, what will happen? The recombinant DNA, it continued to multiply with the foreign DNA or gene of interest. It will get incorporated with the genes that is present in the bacterium, and along with that, the host cell genome, this genes what we have transferred using the plasmid, it is also joined with that and it multiply. So the number of colonies, it increases in more number, so much more identical copies or rDNA fragments, it will be grown into the organisms, grown into the host organism. So this process, it is called as molecular cloning or gene cloning. This process is known as molecular cloning or gene cloning. This is how the process is done. Now just I'll show you how the gene which are responsible for formation of human insulin, it is produced. So what we have to do is, you have to take a insulin gene from the human cell. You have to identify the fragment that is responsible for formation of insulin gene, and that it will be inserted into the plasmid. So we have to cut the plasmid by using restriction endonuclease enzyme, and then it will be joined. After joining, this is called recombinant DNA. This recombinant DNA contains the genes that are responsible for formation of insulin. So this recombinant DNA, it will be inserted into the host cells. So here in the host cell, it starts producing multiple numbers. So what will happen? The same bacterium, after obtaining the genes, the recombinant DNA, the recombinant bacteria, this bacteria is now it is called recombinant bacteria, because this bacteria, it contains human insulin producing gene. So this recombinant bacteria, it now develops the ability to produce the human insulin. This is how, through genetic engineering or recombinant DNA technology, we are producing the human insulin. Now we could talk about the methods of gene transfer. How the next topic is about the methods of gene transfer. The methods of gene transfer. In the methods of gene transfer in plants, there are two methods are there. One is the direct or vectorless gene transfer. The second type, it is called indirect or vector mediated gene transfer. Here in one type of transfer, they are using the without vectors like chemicals, microinjection, electroporation, bioelectrics methods, and liposome mediated method. And the second type, they are using the indirect or vector mediated gene transfer. That is, they are using TI plasmids which is from the Agrobacterium tumefaciens. They are using it as vectors to transfer the recombinant DNA from one to the host cells. Now I am going to explain to you about what are the methods, the various methods are there. Those methods I will explain to you. one by one. First, let me explain about the chemical mediated gene transfer method. In the chemical mediated gene transfer method, they are using two chemicals. One is the fusogenic agent, that is called PEG, polyethylene glycol. They'll use the polyethylene glycol, and also dextran sulfate. What they'll do, they'll take the host cell, along with that they'll take the DNA, recombinant DNA, that is plasmid. Both it will be placed in the same medium which contains the polyethylene glycol or dextran sulfate. So these two are actually fusogenic agent. So what these two things it will do, it will join the protoplasm of the DNA, protoplasm of the host cells, and as well as it joins the recombinant DNA. It connects both. So what will happen, easily by protoplasmic fusion method, with the help of the polyethylene glycol, the DNA it will be taken inside the host cell. This is one method. This is called the chemical mediated gene transfer method. And second method, it is called micro injection method. In the micro injection method, the DNA it is directly injected, or the nucleus or the nucleus which is supposed to be transferred, or the fragment of the gene, it will be directly inserted into the host cell with the help of a fine tip needle or a micropipette. They'll take the host cell, and with the help of this micropipette, they'll take the host cell. With the help of this micropipette, they will inject directly the nucleus into the host cell. So this is the large size pipette. This is called holding pipette. The whole process it is done on a slide, a solid support which contains the agarose on the microscopic slide. So agarose it will be there. They'll have a holding pipette, and the cell it will be the host cell. With the help of a micropipette, they will inject the nucleus directly into the host cell. This is called micro injection process. And third step, it is called electroporation method. Electroporation method. Electroporation method or electroporation method of gene transfer. So here, usually what will happen, we can see that the cell, this is the cell contains the plasma membrane. So this plasma membrane, we know it is selectively permeable membrane. In order to transfer the genes, into the cell, what they'll do, a pulse of high voltage is applied to the protoplast or a cell or a tissue. When a high voltage is applied, what will happen? It makes a pore in the cell. Can you see it here? The cell it will be like that. This is before pulse. During pulse, once the electric field, high voltage electric field is applied in the cell membrane, it creates the hole, pores it will be created. With the help of those pores, they will inject the DNA, into the cell. And then the cell heals with genes or drug inside on its own. This is the electroporation method of gene transfer. The next method, it is called liposome mediated method or gene transfer. Liposome mediated method of gene transfer. The liposome mediated method, it is a artificial, in this method, artificial phospholipid vesicles are used in gene transfer. So you can see that this is a phospholipid. This is a phospholipid, we call it as liposomes. These liposomes are positively charged, and DNA, the DNA it has to be transferred, it is a negatively charged. So what will happen? The DNA plus liposome, they will form a lipoplex. And this this lipoplex, it reaches the cell membrane, the gene or DNA, it is transferred from liposome into the vacuole of the plant cells. This is transferred or carried by an encapsulated DNA into the vacuoles. So what will happen? The DNA that is introduced through the lipoplex, there are a lot of chances for them to get damaged by the acidic pH. And also the cytoplasm of the cell contains the protease enzymes. Those protease enzymes, it can digest the proteins that is present in the DNA. In order to avoid that, the encapsulated DNA, it helps them to protect them itself from the enzymes as well as acidic pH. The liposome, that is the phospholipid vesicle, and the tonoplast is the outermost layer of the vacuole. It fused, the fusion that result in transfer of the genes, that is called lipofiction method of gene transfer. The next method, it is called biolistics methods of gene transfer. This is also called as gene gun method. This is called as gene gun method. This is called as gene gun method of gene transfer. In the gene gun method, the foreign DNA, it is coated on the surface of minute gold or tungsten particles. Minute gold or tungsten particles like 1 to 3 micrometer, it will be there, and that it will be bombarded into the target cells. This is actually like shooting with the help of piston. The same process, they will bombard the cell. It's called gene gun barrel, where they load all the DNA that has to be transferred. It will be coated with gold particles or tungsten particles, and it will be directly bombarded into the target cells. The target cells, it just take up the genes that are transferred inside. This method is also called as biolistics method, gene gun method, microprojectile gun, or shotgun method. The bombarded cells or tissue, while culturing on selected medium, they'll become the transformed cells because they'll take up the gene that is transferred through the gene gun barrel. That's why it's called biolistics method. The next method, it is called indirect or vector mediated gene transfer. In the case of plants, the gene transfers are mainly done through the plasmid vector, that is from the TA plasmid, that is from the Agrobacterium tumefaciens. Whereas the vector, that is plasmid, that is taken from E. coli bacterium, are widely used for both animals and plants. But exclusively for plants, nowadays they are using only the TA plasmid, tumor inducing plasmid, which is from the Agrobacterium tumefaciens. This bacteria has a large size plasmid to transfer the genes through infection process, and a portion of it transferred as tDNA. It's called transfer DNA, and it transferred to the plant genome by like in the infected cells and causes plant tumors. That's why it's called TA plasmid. TA plasmids are mainly when the genes, the TA plasmids are transferred into the, it causes infection in the plants, thereby it causes the crown gall disease, the tumor, the plant tumors. And the same bacteria, it has the ability to transfer the segment of gene into the plant genome. So while infection, they'll also transfer this gene into that. That's why the Agrobacterium tumefaciens plasmids are known as natural genetic engineer of plants. That's why the TA plasmids are known as natural genetic engineer of plants. Next, we are going to talk about the process of recombinant DNA technology. What are the various processes that are involved there after the break? So we are going to have 5 to 10 minutes break. Just relax yourself. After the break, I'll come and explain you about the various process of recombinant DNA technology. Children, if you have a doubt, you can post your doubts now. Welcome back, children. Now we'll learn about the process of recombinant DNA technology. The process of recombinant DNA technology. So let us start the topic. First, let me explain about what are the process of recombinant DNA technology. We know what is recombinant DNA technology. It is a manipulation of DNA, altering the DNA from the DNA or gene of interest from one organism and transfer that to the genome of the another organisms. That's what recombinant DNA technology, are also known as genetic engineering process. And here this genetic engineering process, it involves introduction of foreign gene, introduction of foreign piece of DNA into the genome, and similarly embedding the desired gene into the genome of host cell. One is transferring that, second one is embedding that gene or embedding the desirable gene by the host cell. So this process of recombinant DNA technology, it involves various sequential steps. Some of the sequence steps, it involves isolation of DNA, fragmentation of DNA, amplification of gene of interest, ligation of DNA molecule, insertion of recombinant DNA into the host, obtaining the foreign gene product, and downstreaming process. These are all the 7 important sequential steps that are involved in the process of making the recombinant DNA. First, let me explain about the first step. The first step, it is called the isolation of the gene. First step is isolation of the gene. So what is this isolation of the gene means? In the first step, this is the first step in recombinant DNA technology, where we have to take out the desired gene in a purest form from the organism from which we are going to transfer the gene, or from the donor. Isolation of the DNA in a purest form, that is the first step. The normal cell, if you are going to take the DNA, it not only contains the DNA, but also it contains other macromolecules along with that. It contains RNA, polysaccharides, proteins and lipids. And in order to take the DNA in a purest form, we have to use lot of enzymes so that we can get the purest form of DNA. To take out all these macromolecules, first they will use lysozyme. This is an enzyme which is used to break the bacterial cell wall. Similarly, if it is the host cell, it is going to be the fungi or plant, they'll use cellulase enzyme to break the cell wall, because breaking the cell wall is very important task. After that only we can reach the DNA and take out the DNA from the donor cell. And in the fungi, they'll use chitinase enzyme, because the fungus, they'll have the outer enzyme which is made up of chitin substance. Similarly, to remove the RNA molecule, they'll use ribonuclease enzyme. In order to digest the protein such as histones and non-histone protein that is present along with the DNA, they will use the enzyme protease. And after that, what will happen? Ethanol, they'll place, after digesting all these things, they'll use ethanol. When you place that DNA into the ethanol, it precipitates out and it will come out in the form of fine threads. Then you can spool out and take the DNA separately. That DNA, it will be in a purest form of DNA. So this is the first method in isolation of DNA, isolation of genetic material. Second step is fragmentation of DNA. Similarly, by this method we can even take the gene of interest from the donor organism. Similarly, the other easy method, we can take out of the vectors from E. coli bacterium. Also we can use the previous method. Next is the fragmentation of DNA. The isolated and purified DNA, that is spooled out DNA, it will be treated with enzymes that is called the restriction endonuclease enzyme. You know what is the role of restriction endonuclease enzyme? It is known as the molecular scissors. It cuts the DNA into fragments. So I'll use different kinds of enzymes, restriction endonuclease enzyme, to cut the DNA into fragment. And this DNA restriction endonuclease enzyme, they'll have a particular location as a specific site to cut the DNA, to cut the desired gene, which has to be inserted into the vectors. So according to that, the restriction endonuclease enzyme, it cut the DNA at specific sites. They have a sequential specific sites that will be there. Mostly they'll cut in the palindromic sequences A, G, C, T. Then opposite it will be G, C, A, T, like that. Palindromic sequence, they'll cut the DNA at a specific points. And also the length of the DNA, according to length of the DNA, and they'll make the cut at a specific site. That's why it is called restriction endonuclease enzyme, because it cut the DNA at a restriction site. The desired genes and the vectors are cut by the same restriction enzymes, and finally they'll use the ligases enzymes to bind the desired gene to the vectors. That is done separately. So this process is called fragmentation of DNA. And here, for the fragmentation of process, they will use the agarose gel electrophoresis method. They will load the samples here. And one side of the agarose gel electrophoresis will be negative, the other side will be positive. We know that DNA, there are negative charged particles, they'll move towards the positively charged particles. Once you load it, they run these molecules, it will move according to the base pairs what they have, and they get separated like this in separate bands based on the base pairs, based on the base pairs. Similarly, this method, agarose gel electrophoresis method, it is used for both separating the desired gene DNA fragments, and also the same vector also it will be cut by using the same method. So that what will happen, the vector as well as desired gene both can easily fit together. That can be done. Next is amplification of gene of interest. The next process is called amplification of gene of interest. What is this amplification? Amplification means increasing more in number or producing multiple copies from few copies of DNA. Even with the help of a single copy of DNA, we can produce millions of copies. There is an instrument which is called the PCR instrument, the polymerase chain reactor or PCR. It is used to increase multiple copies of DNA sequence by using the enzyme DNAP, that is called DNA polymerase enzyme. So this PCR, it helped to amplify the single copy of DNA into millions copies of DNA. First, let us see how this process is done. Here, this PCR process, it involves three important stages. One is called denaturation, annealing, extension. Denaturation, annealing, and extensions. These are the three stages. Now let me explain that what are the components that are required for the PCR to functions. PCR, it basically works on dermal cyclers. There will be a lot of variation in the temperature in the polymerase chain reactors. Because of that only they can able to produce millions of copies of the same type of DNA from a single copy. From one, we can produce millions of copies by using PCR. That's why it is called gene manipulation process, or it is used to make multiple copies of DNA sequence. So what are the components that are required? The required components, it includes the template. Template means what DNA to be amplified. That's called template. Next is primers. The primers are single stranded nucleotide. They is called as chemically synthesized oligonucleotides, which is the initiator for multiplication of DNA or replication of DNA. An enzyme, the enzyme is DNAP, that's called DNA polymerase enzyme, or it is also called TAQ polymerase enzyme, because they are taken from particular bacteria that is called Derma acutus bacteria, which can able to withstand the different temperature. They are using that. That's why it is called TAQ polymerase enzyme. The DNA sample, and they need primer. Along with that, they also need nucleotides. The 4 nucleotides: adenine, guanine, cytosine, and thymine. And also buffer is used, and PCR tube is also required. These are all the important components that are included in the polymerase chain reactions. First, let me explain about how this polymerase the PCR works. As I mentioned earlier, the PCR, it works on thermal cycles, heating and cooling process. The whole process, it is done in a machine. And also I mentioned there are 3 stages are there, 3 main stages are there: denaturation, annealing, extending. Denaturation, annealing, and extending. First, let me explain about denaturation process. The denaturation process, the double-stranded DNA, it will be separated into 2 single-stranded structure. The double-stranded DNA, it will be converted into 2 single-stranded structure. That process is called denaturation process. Annealing process. Annealing process, in this process, the temperature for denaturation process, the temperature is 98 degrees Celsius. And then the temperature is lowered. That process is called annealing. Temperature is lowered. That enables the RNA primer to attach to the separated 2 strands. We can see that 2 strands are separated. In that, what is attached? The RNA primer is attached. That process is called annealing. And the third process, there will be slight increase in the temperature from 60 degrees Celsius to 70 degrees Celsius. This process is called extension process. When the temperature is raised, what will happen? The new strands of DNA, it is formed by using TAQ polymerase enzyme. So this process is called extension process. So these are the 3 stages are there. One is called denaturation, annealing, extending process. Now let us learn about how exactly all these 3 processes it is done. The 3 stages are repeated for about 20 to 40 times, so that it can double the number of copies each time. The first process, denaturation process, the 2 stranded DNA, the double stranded DNA, it will be separated into 2 single stranded structure. So for this, temperature is about 98 degrees Celsius, or it will be around 94 to 95 degrees Celsius. You can see that this is the typical structure of DNA, where you have the phosphate, the sugar molecule, nitrogenous bases. These are all the nitrogenous bases. These nitrogenous bases, they are held together by hydrogen bonds. In the DNA, the adenine always pairs with thymine, or thymine always pairs with adenine with a double hydrogen bond. Similarly, guanine pairs with cytosine with triple hydrogen bonds. Here, when you keep this DNA in high temperature, what will happen? It results in breaking of the hydrogen bonds. First step is breaking up of hydrogen bonds. The hydrogen bond that is present between the nitrogenous bases, it will be broken. The bond, it will be broken in the high temperature. Because of that, what will happen? 2 single strands of DNA, it will become 2 single strands of DNA. Both these 2 strands of DNA, it act as a template for the formation of new strands. This temperature, that is 94 to 95 degrees Celsius, it will be maintained for about a long time to ensure that DNA gets separated completely. This will take about 15 to 30 seconds. This is called denaturation stage. The next step, it is called annealing stage. In the annealing process, the temperature is lowered from 94 to 95 degrees Celsius. It is immediately cooled to 50 to 65 degrees Celsius. This reduce in the temperature, it enables the RNA primer to attaches to the template of the single stranded, one from 5' direction, other from 3' direction. It get attaches there, and this attachment of primer to single strand DNA template, that process is called annealing. So this attachment is done mainly by hydrogen bonding. The two hydrogen bond between adenine and thymine, and in between the guanine and cytosine, it will be triple hydrogen bond. This actually, the primers are strands of single strands of DNA or RNA sequence. It's about 20 to 30 bases in length. It will be there, and these are all mainly used to make the complementary strand along with the template. And here, for the extension of this RNA primer, the enzyme, the DNAP, DNA polymerase enzyme, play an important role in adding the nucleotide with the RNA primer so that they will become a double stranded DNA structures. And always this RNA primers, it attaches to the opposite strand, one from 5' end, other from 3' end. So the one is called the forward primer, the other one is called backward primer. So like that, it will get attached. This step usually takes about 10 to 30 seconds. And even though the whole denaturation process as well as annealing process takes about seconds, but they will be repeated for about a minute, for a few minutes, 30 minutes like that, in order to produce large number of copies. And third process is called extending stage. Extending, it is done by slight increase in the temperature from 50 to 65 degrees Celsius to 72 degrees Celsius. This is the final step in PCR process. When the temperature is increased, what will happen? The TAQ polymerase enzyme, that is the enzyme that is extracted from Dermis aquaticus. This is a particular bacterium which lives in extreme temperatures. That bacterium, from that they will extract the DNA polymerase enzyme. That's why it's called TAQ polymerase enzyme. This enzyme, it is used to extend the strands of DNA. 70 degrees Celsius is the optimum temperature for TAQ polymerase to attach the nucleotides along with the DNA, along with the primer, and it adds on the DNA bases, that is nitrogenous bases, by adding one by one to the single stranded in 5' and 3' directions. And this three process of dermal cycling are repeated for about 20 to 40 times. Continuously it is repeated to produce lot of copies. If you want to have more copies, the same process it will be repeated again and again, so that we can get millions of copies of same gene of interest. And here the new fragments of DNA that are made by the PCR reaction, it serves as a template. So how many number of DNA are producing, all those DNA can act as a template and they can produce the same type of DNA again and again. That's why, as I said, from one DNA we can make millions and copies of same type of DNA by using the polymerase chain reaction. And here in a short span of time, short period of time, we can produce millions of copies of same type of DNA. That is the main uses of the PCR or polymerase chain reactor, which are used to manipulate or which are used to produce millions of copies of DNA in a short span of time. Once the thing is, we will get the DNA gene of interest. Next process is called ligation of DNA molecules. The purified DNA and the vector of interest they can be cut by using the same enzyme, restriction endonuclease enzyme. And after that, what will happen? The joining of the fragment takes place. Joining of the fragment of gene of interest and the plasmid, it is done by an enzyme called DNA ligase, and this process is called splicing or ligation process. And after that, the hybrid DNA it is produced. Once the hybrid DNA is produced, the next process is insertion of the recombinant DNA into the host cell. That is the next process, where we have to insert the recombinant DNA into the host cell, and that process it is called as transformation, where you have to take the host cell. The host cell, three species they have used it. One is the E. coli bacterium, the other one is Bacillus subtilis, and Streptomyces species are used. So any host you can use it. Now we can see that we have to take the host and insert the recombinant plasmid. Now this becomes a recombinant organisms. The bacterial cell take up the foreign DNA easily, or else we can use, there are different methods are there to get to insert those recombinant plasmid into the organisms. And now during transformation, the recombinant DNA, it contains the gene for ampicillin resistance is also transferred into the recipient, so that the E. coli will not reject the plasmid or recombinant plasmid. Before itself, they will make the host cell competent to accept the recombinant plasmid. That is the ultimate aim. And here, this ampicillin resistance, the ampicillin resistant, it will make the host cell to be competent to accept the recombinant plasmid, and that's why even in the plasmid itself we have ampicillin resistant genes, are selectable markers are present in the plasmid itself to make the host cell competent enough to accept the recombinant plasmid. Next is obtaining foreign gene product. So now we have seen that how manipulation is done, how multiple copies are produced, and how the genes are inserted into the host cells. And the ultimate aim here to produce more number of desired product. The product may be a protein, an enzyme, a product may be an insulin, a product may be a secondary metabolites, anything it can be there. That is called as that process of producing multiple copies. It is known as to produce multiple copies of DNA, they are using the PCR, whereas to produce more product in large volume, we are using bioreactors. These bioreactors, in the bioreactors, they'll put all the host cells, so that they can produce large number of copies, and also they produce a desired protein in large quantities. For large scale production, they'll use bioreactors. So bioreactors, with the bioreactors we can produce about 100 to 1000 liters of cells can be cultured, and through which we can produce many liters of desired proteins. And here the bioreactors should contain certain criteria, optimum conditions like optimum pH, optimum temperature, it has to be there. Oxygen level, it has to be maintained, and similarly the vitamins, the required vitamins as well as the cultured cells, it has to be there. And when you provide all these things, they will produce specific proteins or enzymes. That is about the bioreactors. And the bioreactors, it has certain components in that. One is the agitator system. You can see that this agitator system is the motor which it will automatically rotate. The rotation, that is called the stirring process. That stirring, it mixes the components evenly, contents evenly, and also that rotation itself produces lot of oxygen for them to grow and react. And oxygen delivery system, it has to be there. This air system, this is the oxygen delivering system to introduce air into the growth, the bacterial growth. and foam control system is also, it will be there to control the foam formation within that. And then temperature control system, it will be there to maintain the optimum temperature. Temperature control system is also there, and also pH control system, it will be there. And there are sensors, there are sensory probes are there. These are sensory probes, it can able to maintain the optimum conditions required for the bioreactors. And also have sample ports, it will be there. Sample ports are mainly to take little bit of samples and check whether the product is obtained or not. This is how it is done. And the final process is called downstream process. With the help of the bioreactors, we can get the product, and the final process, it is called downstream process. The downstream process, the product, it will be subjected for many testing before it will become a finished product, or before it will be released into the market. There are a lot of testing it will be done. The process of testing, it includes the separation and purification process. The separation and purification process altogether, it is called downstream processing. They use selectable preservatives, they'll use it. And also once they get the product, it will be tested for many clinical trials. If it is a drug, or if it is going to be a vaccine, or interferon, insulin, whatever it is, a clinical test, it will be done. After once it has to clear the clinical test, then only the product can be sold in the market. And also quality control testing, it will be there. The quality control testing for each and every product, it varies from product to product. So altogether, downstream process includes separation and purification, formulation with suitable preservatives so that it can be preserved for longer time, at least a year, and clinical trials they'll conduct to test the efficiency as well as the safety of the product if it is going to be human beings, and quality control tests. These are all some of the downstream process. So with this, we'll come to the end of biotechnology. Next topic we are going to talk today, it is about applications of biotechnology. Now we'll go for a 5 minutes break, and after the break I will start the applications of biotechnology. If you have doubts, children, you can post your doubts here. Children, next topic what I learned today is about applications of biotechnology in health and agriculture. Applications of biotechnology in health and agriculture. So first let us see about how biotechnology is used in various fields. Biotechnology are mainly used in the field of agriculture. The field of agriculture, that is mainly in crop improvement. In crop improvement to make a genetically modified organisms with disease resistant plant, drought resistant plant, heat resistant plant, pest resistant plant, high yielding variety of plants. Like that, different types of plants can be produced by using biotechnology. And biotechnology, by using, we can get lot of genetically engineering products which are mainly used for healthcare and pharmaceuticals, secondary metabolites, enzymes, vaccines, insulins, and also biotechnology is used for various therapies like gene therapy to identify various diseases. Biotechnology play an important role, and also the fermentation technology for fermentation processing of various fermented products, and food innovations for therapeutics, and also to treat the sewage and also manage the environmental energy and environmental management. Biotechnology play an important role. These are all the various roles played by the field of biotechnology, mainly the aspects of biotechnology to produce a good product for the welfare of human beings. That is the ultimate aim of biotechnology. So biotechnology nowadays, it is there in various fields of therapeutics, diagnostic, food processing, sewage treatment, energy production, bioinformatics, production of healthcare and pharmaceutical products, genetically modified crops. These are all the various aspects of biotechnology. And first let me explain about the applications in medicine, the field of medicines. The biotechnology or genetic engineering mainly used in production of recombinant human insulin. The human insulin nowadays it is available in the market in the form of inulin. The human insulin, it is the human insulin. Insulin is a hormone that is produced from the pancreas. The pancreas, it has a patches of cells called highlights of Langerhans. In that highlights of Langerhans, the beta cells, there are 3 cells are there. The beta cells of highlights of Langerhans, which is there in the pancreas, it produces the human insulin. The human insulin, it has 51 amino acids. There are 2 chains are there, A chain and B chain. A chain contains 21 amino acids and B chain contains 30 amino acids. So this 21 and 31 amino acids combined together, and human insulin contains 51 amino acids are there. And this A chain that contains 21 amino acids, B chain contains 30 amino acids. They are joined together by disulfide bonds. This insulin that is produced in the highlights of Langerhans, which are mainly used to control the glucose level in the body. The glucose level in the body, it is maintained by the 2 hormones, insulin and glucagon. And for utilization of glucose and release of energy, the recombinant human insulin play an important role. That is the applications of recombinant insulin, human insulin in the field of biotechnology. And this insulin that we obtain from biotechnology, it is used to treat the disease called diabetes mellitus. The deficiency of insulin hormone in the case of human beings, it can lead to a disease called diabetes mellitus. The progressive loss of beta cells due to genetic factors or high glucose molecule or high free fatty acid, it results in impaired glucose intolerance in the body. So that what will happen, high level of glucose it will be there in the blood, which can lead to a disease called diabetes mellitus. To treat the diabetes mellitus patients, nowadays we are using the human insulin which are produced through recombinant DNA technology. So continuous production of insulin is required to treat the various diabetic patients. That is how the insulin play an important role. And let us see how this insulin is extracted or insulin is produced in recombinant DNA technology. So they'll have the human cell. From that human cell, they'll take the insulin gene that is responsible for formation of insulin, and from the E. coli bacterium, they'll take the plasmid, and that plasmid along with the human insulin gene, it will become a recombinant plasmid. This recombinant plasmid is used in the bacteria, inserted into the host cell bacteria. So bacteria are allowed to culture. So these bacteria, once they are allowed to culture in bioreactors, they can produce lot of insulin, because the bacteria are incorporated with human insulin gene. So they produce lot of insulins, and those insulin can be extracted, and after downstream process it will be used for human treatment. But the insulin what we obtain through recombinant DNA technology, it has 3 chains of insulin. Usually what will happen, the human insulin contains A chain and B chain. A chain contains 21 amino acids, B chain contains 30 amino acids. Whereas here the third chain, a linked third chain, C chain, is also present while in the production of human insulin. In the later sequencing process, that is after translation process, the C chain it get excised or removed from the process, and only the A chain and B chain of polypeptides are used in the extraction process, and they are used as human insulin. Now in the brand name of Humulin, it is sold in the market, and insulin was the first ever pharmaceutical product of recombinant DNA technology which are allowed to use it for human beings. Mainly it is the patent as well as the approval was given by the US government. They are the first country to use the human insulin, which is in the form of Humulin. And right from 1982, the recombinant insulin for diabetic treatment, it is given there, and in the trade name it is called Humulin. Next is the human growth hormone. There is in the pituitary of human beings, there is an hormone which is produced from the adenohypophysis, that is called human growth hormone. This human growth hormone is the one which is responsible for the growth in the organism. Sometimes if there is a deficiency in the human growth hormone, it can lead to a condition called dwarfism. The human growth hormones are somatostatin and somatotrophin. So this human growth hormone nowadays they are also produced through recombinant DNA technology. This peptide hormone, which is from the pituitary, they help in growth and development of the organisms. Mainly they are involved in formation of amino acids, uptake of amino acids, and production of proteins. And deficiency of this hormone can lead to a condition called dwarfism. For dwarfism, it could be treated by injecting human growth hormones extracted from the human pituitary glands. The same way how we have produced the human insulin, the same thing it can be done here, where they have to take from the human cell, they have to take the gene for human growth hormone, and from the bacterium, E. coli bacterium, they have to take the plasmid. From the plasmid, this bacterium, this sequence of human growth hormone, it will be inserted, and it made the recombinant DNA. That recombinant DNA, it will be inserted into the host cell, like 3 species. Do you remember that? E. coli bacteria, Bacillus subtilis, Streptomyces species. So they can incorporate that genes, the recombinant DNA into the host cells, and they can produce the human growth hormones. The human growth hormones, mass production of human growth hormones, they are using fermentation technology, like they are using large bioreactors for production of large scale of human growth hormones. And this human growth hormone somatotrophin, it is nowadays used to treat the growth disorders in the case of children. Next is the human blood clotting factor 8. This is also can be produced, and similarly interferons, recombinant vaccines can also be produced by by the recombinant DNA technologies. So now the human blood clotting factor 8. This human blood clotting factor 8, it is located in the X chromosomes. The X chromosomes. The genetic defect in the species, if there is a defect in the blood clotting factor 8, it results in a disease called hemophilia. In the hemophilia patients, once there is a cut in the blood vessel, the blood oozes out, and after some time what will happen, the blood clotting, it will be there. Once the blood clotting is there, the blood will not oozes out. For the hemophilia patient, the clotting time, it will be prolonged. Usually clotting time, it takes about less than a minute or maximum 2 minutes. Whereas in this case, they will be having a prolonged clotting time, and also internal bleeding, it will be there for hemophilia patients. This is mainly due to the clotting factor 8, deficiency of clotting factor 8. So here, through recombinant DNA technology, the clotting factor 8, it is isolated from the blood of normal human beings and used for treatment for hemophilia patients. And through the recombinant DNA technology, large production of recombinant factor clotting factor 8, it was produced with the help of the Chinese hamster ovary in the baby hamster kidney cells. From that, they are producing the blood clotting factor 8, which are used to treat the hemophilia patients. Similarly, interferons are antiviral proteins. Antiviral proteins which are used to treat various viruses, viral infections. So these interferons are antiviral proteins which are first discovered by Alec Isaacs and Gian Linderman in the year 1957. And the interferon, they are classified into alpha, beta and gamma interferons. And the role of these interferons, once the cell is infected, the interferons, it will be produced. This interferon that is produced from the infected cell, it inhibits the viral replication and protects the cells from further damage. For a cell, if it has to produce an interferon, the cell has to be first infected with virus. After infection only, the antiviral proteins are formed. Those antiviral proteins that are interferons are formed by the infected cell that inhibits the viral replication and protect the cell. That's the reason why any viral infections or viral fever, it lasts for about 14 days. So these interferons can also be produced by recombinant DNA technology. But the organism what they are used here, it is the yeast, Saccharomyces cerevisiae, that is used in the production of recombinant interferons. So these interferons can be used to treat various viral diseases like cancer, which is caused by oncogenic viruses, AIDS, and multiple sclerosis, hepatitis C, and Hep zoster. And production of interferons and the commercial cost of interferon is very high. That's why these interferons are not within the reach of common man due to high cost in the production that is involved in production of interferons, whereas the other metabolites are not that much high cost when compared to the interferons. Next is the recombinant vaccines. Recombinant vaccines can also be produced through the recombinant DNA technology. Through recombinant DNA technology, we can produce lot of vaccines. The vaccines mainly used for some of the examples are HBsAg vaccine and HPV vaccines. Now let us learn about what are the various recombinant vaccines are there. There are 4 different types of vaccines are available. One is called the subunit recombinant vaccines, attenuated recombinant vaccines, and DNA vaccines, and vector vaccines. These are the 4 different types of vaccines are there. First, let me explain about the subunit recombinant vaccines. The subunit recombinant vaccines. Children, just copy this flow chart. This flow chart and this one. I'll give you 2 minutes time, and after that I'll come and explain about the different types of recombinant vaccines. Okay, children, we'll continue the topic. The next is about the subunit recombinant vaccines. Here in this vaccine, they use components of pathogenic organisms. They're not going to use the whole organism as such. They'll use the components. It may be a macromolecule. From that, it may be a protein or a peptide or DNA of the pathogenic organisms. Like that, a part of the organism is used. That's why it's called subunit recombinant vaccines. And here, the advantages of these vaccines, it includes their purity in preparation, stability and safety to use, because we are not going to use the whole organism as such. It's going to cause the infections, but here we are going to use certain extraction, certain components from the organism, pathogenic organism. That's why it is called subunit organism. It may be a protein or a peptide or a polymer or DNA of the pathogenic organisms. This is called attenuated recombinant vaccines. These are all genetically modified pathogenic organisms that are made non-pathogenic. Here the same pathogenic organism, it is made non-pathogenic and used as vaccines. And this recombinant DNA technology, through genetic engineering process, they will make the organism non-pathogenic and use them as live vaccines. These vaccines, it will only create antigenic response in the organism, but it will not cause any infection to the organisms. Next is the recombinant vector vaccine. Here we are using, instead of delivering DNA or mRNA directly to the cells, they are using vectors from the bacteria or harmless viruses. They will use carrier to introduce the genetic material into the cells. The recombinant vector vaccines are mainly to protect animals from infectious diseases. Then we have the DNA vaccines. These are all DNA vaccine, immune responses of the body. It is stimulated by the DNA molecules. So they are using DNA as a vaccine. A DNA vaccine, it mainly consists of genes encoding an antigenic protein. Gene encoding an antigenic protein inserted onto the plasmid and then incorporated into the cell. So here the plasmid used as a vector to transfer the DNA into the organism or the target animal. So this DNA which enters into the body, it it gives instruction and make antigenic molecules, so that those antigenic molecules, it provoke the antibody response within the organisms. And this DNA vaccines are relatively very easy and inexpensive to design and produce. So many copies can be produced by using PCR and also by using the bioreactors and downstreaming process. These are all about DNA vaccines. So now we are going to explain about the production of recombinant HB vaccine. For that, they will take the HB antigen producing gene and introduced into the plasmid. Now it becomes recombinant DNA. This recombinant DNA, it will be inserted into the host cell. The host cell, as I mentioned earlier, it is the Saccharomyces cerevisiae, the yeast. And that it will be introduced into the host cell, again the yeast. So the host cell, from the host cell, they can produce the HB vaccine. These HB vaccines, hepatitis B vaccines, it is used to treat the various hepatitis diseases. This is how recombinant HB vaccines are produced. Now I want to explain you about the edible vaccines. Instead of taking the vaccines in the form of injections or orally, now we have edible vaccines are there. The edible vaccines, the selected genes are introduced into the plant, and the plant itself produce a product. That product itself contains it is coded with certain protein that induces the immune response. Edible vaccines are mucosal targeted vaccines which are used for both systemic and mucal immune responses. Mainly these edible vaccines are used to treat measles, cholera, foot and mouth disease and hepatitis. That is called edible vaccines. And these are some of the products, recombinant DNA products that are used nowadays. Okay children, so with that I'll stop today. Just copy this slide and next class I'll explain you about the various applications of the genetically modified products and their applications. Okay children, we'll see you in the next class. If you have doubt for another 5 minutes, it is open for discussion. You can ask your doubts. Thank you very much children.